This experiment will help us see how plants exchange gases through the process of photosynthesis. We will really be focusing on carbon dioxide and oxygen. We're going to use Bromothymol blue, or BTB, to help us see this gas exchange. Bromothymol blue solution can indicate the presence of carbon dioxide in water. When there is little or no carbon dioxide present, BTB will show a blue color. Depending on the amount of carbon dioxide, BTB will change to either green or yellow. A greenish yellow indicates that more carbon dioxide is present. I have 30 milliliters of water in this beaker. Now I need to add 1 milliliter of bromothymol blue, or BTB, and I have to observe the color change. To add the BTB, I am going to use a pipette so that I can precisely add 1 milliliter of bromothymol blue to the water. I place the beaker of water on a white background so that we can easily see the color change. In my pipette, I have 1 milliliter of BTB. Note that it has a very dark green color to it. Now I'm going to add it to the 30 milliliters of water in the beaker. Note the color change. The BTB mixed with the water now has a bluish tint to it. Before I can begin adding CO2 to the water and BTB solution, I need to make a control test tube. So I'm going to pour 10 milliliters of the solution into a test tube and cap it closed. Now that I have my control test tube set aside, I can begin the experiment. I am now going to add carbon dioxide to the solution. In order to do that, I am going to use a straw and I'm going to gently blow bubbles into the solution. Think about what gas is being exchanged when I am blowing into the beaker. Also, pay attention to the change in color. Right now, the solution has turned to a light green color. I'm going to continue to blow one more time to see what will happen to the color. Now the solution is more of a greenish yellow. This is the solution that we are going to use and pour it evenly into our two other test tubes. I have now added 10 milliliters of the different colored solution to each of the two remaining test tubes. I am now going to cap one of them to keep it as my control. This control will show the carbon dioxide rich solution compared to the two other test tubes. Now that I have both of my control test tubes capped, I can begin the last part of this experiment. In the remaining test tube, I need to add a sprig of Elodea. Elodea is a common exotic aquarium plant that has also escaped into some local waterways. Elodea is an excellent plant for our studies of photosynthesis because it is easy to grow and readily available. Recall from earlier discussions and studies that photosynthesis requires certain raw materials and produces certain products, one of which is released by the stoma as a gas. Our goal is to add Elodea to the remaining test tube so that we can see how the colors change after sitting for 24 hours. The results will show us how gases exchange through the process of photosynthesis. On the petri dish is a sprig of Elodea. I am now going to add it to our third test tube. I'm going to put it in so that the stem is facing down so that the process of photosynthesis can occur properly. It's important that the Elodea is submerged under the solution so that it can allow the process of photosynthesis to occur properly. Now that the Elodea is inside of our third test tube, I'm going to cap it closed. All three of our test tubes are now capped closed, so now it's time to move on to our last step of the procedure. Our final step is to place all three test tubes in front of a heat lamp. The test tubes will remain in front of this heat lamp for 24 hours. We will come back and check our results after it has been 24 hours. It has now been 24 hours. The test tubes have stayed in front of this light for the entire time. Now, let's go ahead and observe the changes. Here's what the test tubes look like after sitting in front of the light for 24 hours. In order to see the test tube with the Elodea better, I'm going to turn it over so that we can just focus on the color. Note that test tube 1, which was our control, is still the blue that it started as. Also notice that our second control, the one that was carbon dioxide rich, is still the greenish yellow that it was when it started. But if you look at the test tube with the Elodea in it, we can see that there has been a color change. We added Elodea to the test tube with the carbon dioxide rich solution. After 24 hours, the solution has started to turn back to green. What does this color change mean? What do you think would happen to the color solution if we let it sit for another 24 hours?